Vazquez, and I will be talking today about the phylogenetic relationships between the members of the genus Notropis and the family Cyprinidae. The family Cyprinidae is actually one of the largest freshwater fishes families in the world. It includes 371 genera and over 3,000 described species. Members of this family occur in North America, Africa, and Eurasia. The genus Notropis is a wholly North American genus. They are commonly referred to as the eastern shiners. They're a very speciose genus with members ranging from Canada to Mexico. In the past, Notropis was actually a much larger genus with many subgenera present within it, as you can see from this figure down here. Maiden in 1989 was able to place Notropis and its allies into the open posterior myotum clade, which is a clade based on morphological features. However, in the same paper, Maiden actually removed 4 subgenera from this genus and elevated those 4 subgenera to genus level. And these include Lythorus, Luxellus, Cyprinella, and Notropis, allowing the genus to actually be contracted and become much smaller than it was then. Currently, there are 91 extant species of the genus Notropis. 4 species are currently extinct and 1 subspecies as well. These include Notropis orca, Notropis sinusinus being the subspecies of Sinus equentis, and Notropis aleodon, Notropis amechii, and Notropis saladonis. 15 of these species are currently red listed by IUCN and also have some species listed in the United States in various fashions, including at the state level and at the federal level. The current genus is still much contested by Cyprinidae systematis, and with many wanting it to be split up further. There are currently 4 groupings within the genus Notropis. This includes the subgenus Alburnos, the subgenus Hydrophlox, the subgenus Notropis, the Mexican minnow clade, and the rest have either undetermined group designations or have no determined sister taxa. These 4 groupings are based on morphological characteristics, mitochondrial markers, and nuclear markers. In 2006, Maiden et al. Was able to complete a very large phylogeny for the genus, and I'm going to be discussing that later. So the most complete phylogeny for the genus was made by Maiden et al. In 2006. The phylogeny was made using cytochrome B sequences. Their analysis used members of the genera in the OPM clade, the open posterior myotum clade, of North American cyprinids to best determine if the genus Notropis was monophyletic. They used both Bayesian analysis and maximum parsimony analyses for building their phylogenies. Maiden et al. Also was able to determine that the genus is actually polyphyletic, and with this also described 3 new genera from the species Notropis. All right, so you can see this is a zoomed-in picture of the tree that they were able to develop, redrawn from the actual paper. As you can see, the arrows designate Notropis species, and you can see that it is actually very dispersed throughout this clade here. Notropis with quotation marks refer to species that will have no current designation, at least by Maiden et al. Standards. Some of them are actually pointing to genus that aren't Notropis, obviously, but these are some of the genera that Maiden et al. Was able to elevate from Notropis species. And these clades include the subgenus Alburnos. They elevated it to actual species or genus level. Miniellus, which is a small genus. Hudsonius, another small genus that they assume to be a basal group to the remainder of the Notropids. This Notropis on Urosaurus clade. And finally, this clade right here is the genus Notropis, at least how they view it. So they were able to contract the entire genus down to only a few species compared to it being a 91 species clade. So the purpose of my study was to utilize more data than what they had and determined if the species tree that they developed in 2006 is still valid today, or if there's better consensus for the certain associations that they found. The reason I'm also doing this is to find new relationships between species that can be utilized by conservation biologists to find surrogates for species of conservation concern. To do this analysis, we used publicly available data. We built phylogenetic trees to best understand the relationship between the species in this genus, Notropis, and the allies in the OPM clade. So we searched and accumulated publicly available sequences from GenBank for species for Notropis species, members of the OPM clade, and also utilized Rhaenictes atritulis as the outgroup, as it is part of the western clade of the North American Cyprinidae. The three most speciose gene sequence or genes within GenBank included CyP, CO1, and RAG1. There are actually lower numbers of individuals as you go from CyP or from left to right in the genes. We aligned the sequences with muscle, used J-model tests to find the best model of evolution for each individual gene. We used partition finder when we concatenated the genes to find our best way to partition our data for analyses. And then we used maximum likelihood analysis using RAXML to make the phylogeny. The model evolution that we utilized was GTR plus G. We started with 10 random trees and had 1,000 bootstrap replicates for the branch support. The generated species trees were then compared to Maiden's tree in 2006. So this is my complete concatenated tree. These colors are associated with either designations that Maiden et al. In 2006 had, or their species that were actually included that were not included in their own phylogeny. So I'm going to zoom in on two different parts. So the first part being the top part, as you can see, the Notropis is basically strewn throughout the entire tree as well. You can see different species that some of them are associated with. Some of their designations actually still are potentially monophyletic, but I will discuss that further in a little bit. And so with this tree, I was able to determine that indeed Maiden et al. In 2006 was correct. The Notropis is a polyphyletic group, and there's two reasons for this. One, the genus Hudsonius that Maiden et al. In 2006 was able to describe was in fact a basal group to the entire Notropin clade. You can see here, this is Notropis cummingsae and Notropis alpinus. They actually are a basal group to the remainder of the Notropids. And so I believe that the way they describe the genus, it should actually be, these two species should be elevated to that genus level. The other factor is the fact that Notropis nasas should actually be removed from the Notropis genus and be included into the Hypognathus genus, which are the silvery minnows. So those are my two instances of why. The remainder of this talk is just basically going to show the differences between my concatenated tree and Maiden et al. 2006 tree. So their genus Notropis that they found was actually not monophyletic compared to the one I had. As you can see that there's certain clades that do show up within the genus, but they are not a monophyletic group. Also, the other clade, the Notropis longistris clade, is also a non-monophyletic group. The genus Miniellus that they described was also not a monophyletic group. The genus Alburnos, described by Maiden, was also a non-monophyletic group. Interestingly though, this clade right here was described in 2011 by Kastner et al. To be hydrophlox. And in their analysis, Maiden et al. Described hydrophlox to be a non-monophyletic group, and they disregarded it, but in fact it actually still shows up as being a monophyletic group. So in conclusion, the genus Notropis is no longer a monophyletic group. Many of the groupings for the genus put forth by Maiden are not monophyletic. There are new clades that that were determined from the phylogeny and need to be named. One of the clades is the Uranoscopus vulcellus, Chihuahua, Oops, and Heterodon clade. The genus Hudsonia should be ascribed to both Altipinus and Cummingsae. Notropis nasla should be moved to the genus Hypognathus. And obviously, we still need more data to determine the remaining members of the group of Notropis to see where they would stand. So what's next? Sequence Cyp B, CO1, RAG1 genes from fishes present in the current, not present within the current phylogeny. This includes Notropis buccola, which is an endangered fish found in Texas. It's federally endangered. Sequences from the Mexican minnow clade, as that clade was not actually incorporated into this phylogeny. And also sequence different genes for species that were missing one or more gene sequences that were not included in this phylogeny. Also utilize Bayesian analysis to determine if the topology that I found with maximum likelihood would actually be similar to it. Use constrained tree analyses to determine if the remaining members of the group are monophyletic, and incorporate other nuclear and mitochondrial gene sequences to get a better supported phylogeny. Some people I want to thank: Haley Brock, an undergrad in my lab that helped me to gather a lot of these sequences, my two co-advisors for helping me with this project, Neil Platt for helping me with the phylogenetic analyses, as well as Jorge Salazar, my girlfriend Rachel for putting up with my stressful ways, HVC, the high performance computing cluster at TTU for some of these analyses, and I use Cypress a lot in this talk as well.